Hello and welcome to Chapter 5, Part 1, Membrane Transport and Cell Signaling. This is EFB103, and I'm Dr. Chris Whips. Let's get started. So here on this slide at the bottom, you see the main topics of this chapter, and we'll go through them, starting with membrane structure. So one of the first things that we get into with cell membrane structure is this idea of what's called the fluid mosaic model. And so what we have in the fluid mosaic model is that we have something arranged in a bilayer. What is it? Phospholipids. And inserted in that lipid bilayer are proteins. So there's that mosaic model, right? So cell membrane is made out of phospholipids as well as proteins. And we'll see other things as well here. So here's a cross section of what a cell membrane might look like. You see, we talked about cytoskeletal elements. You see some of these connected to integral membrane proteins here. Some proteins, like this guy here, are peripheral proteins. So it's not embedded in the membrane, but it's associated with the membrane or proteins with the membrane. We see some little green carbohydrate chains that are either attached to proteins or lipids. And here we see an extracellular matrix. So the cell membrane here is sort of much more complex than just a phospholipid bilayer. But let's start with that phospholipid bilayer. This should be review from a previous chapter. Here's a phospholipid. So we have these fatty acid chains, tails, you can think of them as. Here's that glycerol molecule. When one of the carbons has a phosphate and a choline group attached to it, this makes this end of this molecule very polar. This other end with the fatty acid tails, not polar. And that's how we have this membrane structure formed. So those polar heads will interact with water, so that fluid of life, extracellular, intracellular fluid. And those tails will be excluded from water, sort of turned away from the water, and then therefore they are turned toward each other. Now, this interaction here, it's not that these tails are attracted to one another, it's just that they're pushed away from water, which is on either side of the membrane here, and so they face each other. To demonstrate that, you could do something like this freeze fracturing technique. So if you freeze a cell membrane and then fracture it, where will that membrane break? It's along the middle of it, where really there's no true connection, right? There's no covalent bonds holding those little tails together. There's just a space here. And so the membrane will crack. And you can see that in some of these transmission electron micrographs. Here's a nuclear membrane with all those nuclear pores. We can see that here in another way. We see, this is kind of interesting too. Here's the inside of the extracellular layer. So as we see in this cartoon, here's the intracellular layer. What do you notice? What's the difference? The intracellular cytoplasmic layer, rather, is got a ton more proteins embedded in it than the extracellular one. And you see that represented in this cartoon to some extent. Evidence for this fluid mosaic idea. The fact that these proteins are embedded in the membrane, some might be anchored to cytoskeleton or extracellular matrix, but some of them are just there in the membrane, and then they can move in a fluid manner. And so this concept of a fluid mosaic model. And you can demonstrate that with by setting up two cells and say you have a labeled protein that is embedded in those cell membranes. So here's a human cell and a mouse cell, and we're showing them as blue and red. So we can distinguish them. If you fuse those cells, you don't just end up with a bigger cell that is half got blue proteins, half got red proteins. Those proteins drift and move around in that membrane. So that's that fluidity. The phospholipids move around. Each individual phospholipid isn't connected to the one next to it by a covalent bond. They just cluster together. So they move around in a very fluid way. And I encourage you to watch some of the links to these videos that I've put on the slides. You can download the slides on Blackboard. So what's going to influence fluidity of a cell membrane? Well, we can look at a couple of examples here. Let's look at the tails of those phospholipids. Here we have unsaturated tails. So think back to lipids and fatty acids. That means there's double bonds and they're kinked and curved. And that means they can't squeeze together as tightly. And when they can't squeeze together as tightly, they're not as crowded here in this cell membrane either. And that's going to make the membrane more fluid. Just like, vegetable oil, when we have those unsaturated fats, is more fluid than animal fats, which is like butter at room temperature is solid. So it's similar with the tails on phospholipids too. Here we see very saturated tails on phospholipids. So this membrane is going to be much more viscous, less fluid than this one over here. Another important compound when we think about membrane fluidity is cholesterol. So cholesterol has a bit of a buffering ability on the fluidity of the membrane. So at, say, really low temperatures, that would mean, think of the membrane kind of stiffening up at low temperatures. Cholesterol will help maintain its fluidity so it doesn't kind of get locked up and solidify. But at higher temperatures, it kind of has the opposite effect. So it actually helps reduce membrane fluidity, because if you increase the temperature too high, it becomes too fluid, right? And could kind of fall apart. And so cholesterol will sort of reduce that fluidity at higher temperatures. And that's why we say that cholesterol has this buffering effect. Okay, quiz time. Given the choices, which of the following factors would tend to increase membrane fluidity? All right, so yeah, if you have more unsaturated phospholipid tails, that is going to increase that fluidity. And that's just harkens back to this slide here in panel A on the left here, more fluid when you have unsaturated hydrocarbon tails. All right, what about that other component of the cell membrane, those proteins? So here are some examples. We have integral proteins. So these are completely embedded within the cell membrane. Then we have these peripheral proteins. They're only partly embedded in the cell membrane. The green regions you see are either alpha helices or beta sheets. And then you've got parts that are on the inside and outside of the cell extending out from the membrane. So let me ask you this question. If you look at any of these, or the transmembrane proteins at least, what would be a characteristic that they would all have in common, given the choices here? All right, so yeah, a hydrophobic domain. So this part that's in amongst those phospholipid tails, and it's shown here in blue. So here's the whole alpha helix, for example. Part of it's red, that's the hydrophilic part. And then this blue would be the hydrophobic part. Now, the actual inside part of the, say this is a channel protein, which we'll get to, that might be hydrophilic itself, but where it comes in contact with the membrane, inner part of the membrane here, would be hydrophobic. So what do these proteins do? This is a nice figure from your text showing some examples. Here's a transport protein. So here's like a channel where ions or molecules can pass through. Here's another kind of transport protein that's moving actively compounds from one side of the cell membrane to the other. Here's enzymes. They don't form channels per se, but they are performing some function. Maybe they need to be localized somewhere in a membrane, so they kind of stick together. There's signal receptors. So cells get signals externally, and what receives those signals? Proteins on the surface often. Cells talk to each other, connect to one another. So here, glycoproteins are very important for cell recognition. They may be important for connecting. You've heard about this already, or attaching to that extracellular matrix. So carbohydrates are really important when we think about cell recognition. So how does one of your immune cells know that it's bumped into one of your own liver cells versus some other kind of cell or foreign invader? It has a lot to do with glycoproteins or glycolipids. So when you look at a cell membrane, you might see glycolipids. So imagine a phospholipid with some carbohydrate chain, little carbohydrate chain attached to it, or a protein with a little carbohydrate chain attached. We jump back here. Here's the cartoon version of that glycoprotein, little green carbohydrate chains here. And then here's what it actually might look like, a lot of carbohydrate extending out off of the cell membrane. So that's really important for cells to identify who's who and what is self if we're talking about an individual. All right, so how do these things get there to the surface of the cell? Well, you got to go back to the chapter where we talked about all the different cell structures. If we're talking about a protein, that's going to be built at the rough endoplasmic reticulum because this is an embedded protein. And then you have a carbohydrate, let's say. So here's a transmembrane protein and it gets a carbohydrate attached to it. At this point, it's on the interior of this little vesicle, right? And this is colorized so you can see what's going to end up on the outside. Ultimately, this will get processed in the Golgi apparatus. And then here's the vesicle, same little glycoprotein. And when that vesicle fuses with the cell membrane, what was the inside of that vesicle has now become the outer surface. And here's one of those transmembrane glycoproteins. And then the same would be said for a glycolipid. Here it's produced at the ER and off it goes to the Golgi. And eventually this vesicle fuses with the membrane. And here are those glycolipids with their carbohydrate portion projecting off of the cell. So let's talk about transport across cell membranes now. And a key feature to pay attention to is this idea of selective permeability. What this means is the cell membrane is permeable to things, but not everything. And we'll talk about now what allows certain molecules to pass across the membrane or not. So there's two kinds of transport: passive, that requires no energy, and active transport that requires energy. And then we've broken down passive transport into a few different categories here. So when we think about passive transport, one of the first things you need to understand is this concept of diffusion. If you take this beaker here in this example, you put a drop of dye in the middle of it here, and over time that is going to spread. It's going to diffuse. And what's driving that diffusion is a concentration gradient. So you have an area of high concentration, and those molecules are moving to an area of low concentration to the point where they're evenly distributed and there are no concentration differences. So they're spreading out to basically remove that gradient over time as they diffuse. So formally, we can think of diffusion as the movement of something, molecules usually, in a solvent, from an area of high concentration to low concentration. And that's important to have that complete thought when you think about diffusion. Let's put a membrane in, say, that beaker now. So we have a membrane here in this example, and we have molecules of dye shown in little cartoon dots. And this membrane is permeable to this orange dye here. And so over time, the membrane is maybe going to slow down diffusion a little bit. So, temperature, the degree of that concentration gradient is going to influence diffusion. And the presence or absence of a membrane is also going to influence that diffusion. So this will slow it down. You see a couple, these ones sort of bouncing off the membrane here. But eventually, you'll reach an equilibrium where these are at equal concentrations on either side of that membrane. And that could be true, say, in the second example here, where we have two different kinds of dyes on either side of the membrane. They're both going to work the same way. The concentration gradient of one is, that's what it's following. It's going down its own concentration gradient. And same with so the purple one's going to move this way, the orange ones move that way. And eventually they're going to reach sort of an equilibrium. And that doesn't mean that the movement of these has stopped completely. So this is an equilibrium. So still, okay, orange one moves over here, one moves back. It is still happening in a very dynamic way in this equilibrium. It hasn't come to a grinding halt. Now in a cell, that membrane isn't permeable to whatever you happen to put in there. So some things can pass the membrane. So small non-polar things like gases, oxygen, for example, can cross the cell membrane without any problem because they're teeny tiny. They can squeeze through there. But these channel or carrier proteins are needed to get a lot of other things across the membrane. So big stuff that can't cross the membrane, polar things, These carrier proteins are highly specific, so they'll only let certain things through that can attach to them and pass through. A special case here is a channel protein, which is same as this channel protein up here, pretty much the concept of it. But these are specifically called aquaporins that allows water to pass through. And so we'll talk about that one first when we talk about osmosis. So when we've been talking about diffusion, we're really thinking of water as being kind of just there in the background, right? Water is the solvent. And we've been focusing on solutes, so those dye molecules or a glucose molecule, it's going to move across the cell membrane. But water is also going to work by moving down its own concentration gradient if there are differences in concentration. And so the movement of water from an area of high to low concentration of water is called osmosis. And another way you can think about this is water is going to move toward an area of high solute concentration high solute concentration. So let's take a look at this as an example here. So we've got this U tube, and there's water. You put some water in it, and you have this membrane that separates the two halves. Now this membrane is permeable to water. So here we're getting into that idea of selective permeability. Permeable to water, but it's not permeable to the solute, this green molecule here. It's some kind of sugar molecule. So water can cross this, but not the sugar. If we have a high concentration of the sugar over here, you can think of this as this on the left is much more watery, and this is less watery because you've put more sugar in it. And so water is going to move down its concentration gradient. It might help to look at this image here. You see lots of little water molecules over here, high concentration of water, versus this side, low concentration of water. And because the sugar can't move across the membrane, it's not until you get to the point where the water can pretty much dilute that sugar so the concentration of it is equal on both sides that water will stop moving. Again, this is dynamics, moving a little bit back and forth even when it reaches equilibrium. And so in this scenario, this right side of the tube actually is going to increase in height because the water is moving from left to right to dilute that sugar, or the water moving down its concentration gradient. And I'll say that, I think thinking about the dye molecules diffusing or a sugar molecule diffusing, that's a little easier, I think, sometimes than thinking about osmosis and water moving, because again, you usually think of it as this background thing, but remember, it is a molecule too, and it has concentration gradients, and it's just moving down its concentration gradient. That's osmosis. Some terms that are important when we talk about osmosis are hypertonic and hypotonic. So think hyper is above, and so this is a solution that has a high solute concentration. So think, salty, like the ocean. Well, these are relative terms. So the ocean is hypertonic relative to the water in, say, a river, freshwater stream. Hypotonic has a lower solute concentration. So we can use that same example. That river is hypotonic relative to the ocean. So the river is less salty. And so if you actually had the same solute concentration, that's the last point here, we would say that's isotonic. So iso is the same, hypo is below, hyper is above. And in our cells, water will move through those channel proteins, those aquaporins, again, toward the hypertonic solution. So let's look at how that might work in animal and plant cells. So let's take erythrocytes, your red blood cells. So there's some salt concentration inside of our cells. And if you put them in a fresh water solution, so a hypotonic solution, not salty, those cells will burst because water is going to rush in. Water is moving down its concentration gradient, so into the cell, right? Because there's a lot more salt inside the cell than outside, and it's just fresh water. And so it'll rush in, and actually in the case of this red blood cell, it's going to burst the cell. You get so much water in there. An isotonic solution, so the concentration that you're putting, the medium that you're putting the red blood cells in is the same concentration of what they have internally. They're going to look the same. They're going to maintain their shape. And then the last one would be a hypertonic solution. So this would be a really salty solution, more than what's in the cell. Water will rush out of the cell. And so that cell's going to shrink, right? It's losing volume because water is leaving the cell. Now, this basically the same idea happens in plant cells, but in the case of plant cells, they actually want to sort of be in a hypotonic solution to maintain the rigidity of those plant cells. So they're taking up water. They don't burst like you'd see with an erythrocyte, but that maintains that normal turgor pressure. An isotonic solution, that's when the plant is starting to look a little limp. And a hypertonic solution, that'll cause the plant cell within its cell walls to shrink. And this is called plasmolysis, when that actual cell shrivels up within the cell wall. So this can create, the medium that organisms are living in can create some challenges. So if you take this paramecium, for example, it lives in a freshwater environment. And we've talked about this guy before. It is basically losing or gaining water across its surface all the time. So it uses this contractile vacuole to collect and then pump out that water. Otherwise, it's going to keep expanding and getting bigger until it explodes, like that red blood cell example. Let's take marine fish, for example. So they live in a salty environment. So that means that they're going to be constantly losing water across, say, their gills and skin, for example, in living in that salty environment. So they have to cope with that, and they have evolved different strategies, adaptations to deal with those different environments. Okay, here's a question, sort of put some of your osmosis knowledge to the test. So here's one of these U-tubes. There's a membrane that separates side A and side B. And a key point here is the membrane is permeable to sodium chloride, but not glucose. So glucose can't move across that membrane at all, but sodium chloride can. So you see the starting concentrations here in molarity of both glucose and sodium chloride. And so the question here is, at the beginning of this experiment, what can you say about this situation, given the choices here? All right, so side A is hypotonic to side B because you have more solute over here. If you just look at the concentration of glucose and sodium chloride, there's a little less sodium chloride over here, but there's a lot more glucose molecules on side B than on side A. And so that means side A is hypotonic, again, relative to side B. So what happens over time, though? This is at the very beginning, but what's going to happen over time? Well, we know sodium chloride can move, so it will move down its concentration gradient. So you'll get sodium chloride moving from left to right here, and that'll eventually reach an equilibrium, so the same concentration. But glucose will never change. That will not happen. And so what's going to happen then is water will move down its own concentration gradient from left to right, and that will cause the water level inside B to increase and basically dilute this glucose. So over time, this will become more dilute, and so this concentration will increase. This one will decrease because you're changing the volume on either side, and so that's going to change the concentration of glucose. So let's move some things in and out of cells that are bigger than, say, water, which travels through those aquaporins, or bigger than gases that can actually just cross the membrane. Most things are coming in and out through channels or carrier proteins like we see here. So a channel is really pretty straightforward, right? So it provides a space. This internal part of the channel is typically hydrophilic, whereas these parts that are interacting with the cell membrane are hydrophobic. And the channel may have a certain width or size that is size selective, but for the most part, whatever can fit through that channel is going to pass and go down its concentration gradient, whether that's into or out of the cell. A carrier protein is a little bit different. These will specifically bind to whatever this compound is, orange dot molecule here, and allow that thing to come in or out down its concentration gradient. Again, this is still passive transport. but it's very specific. And so this is called facilitated diffusion. It's facilitated by those carrier proteins that bind to that molecule. We see this a lot with sugars and amino acids coming in or out of the cell. So it's passive, it's down a concentration gradient, but it's specific. It's not just a hole in the cell membrane that anything can pass through. But because it's specific, it is subject to saturation. The example I like to give is, imagine you're going to a sporting event or a concert, and there's, only so many turnstiles or doors to get in, but there's a big crowd waiting to get into that. And so you're limited by the number of carrier proteins that can get in or out down their concentration gradient, whichever way we're talking about. And so it's going to take time, subject to saturation. If you've only got 3 gates here, you can only do 1, 2, 3 things at any given time. Let's move on now to active transport. So this takes energy. That's why this is an active process. So there's 3 key things to pay attention to here. We're now moving compounds or molecules or ions against their concentration gradient. So this is against sort of that passive diffusion. So that's why it takes energy, because this isn't just some process that's going to happen spontaneously. So it requires energy, and maybe not intuitively, but if you think about it, it makes sense. It requires a carrier protein, because without it, this is not just going to happen on its own. It requires energy and something to process that energy. There's a variety of different kinds of transporters. So uniporters that move 1 molecule at a time, symporters move 2 at a time, and then something called an antiporter is moving 2 things at a time, but 1 in and 1 out, or vice versa. So those are the different kind of transporters that we see. And a great example of this is where we see the maintenance of membrane potential. So in our cells, the interior of the cells along the inside of the membrane here has a slightly negative charge, and on the outside is a slight positive charge. And this is due to this electrochemical gradient that creates this membrane potential. This is really important in nerve signal transduction. And it's maintained or built or created, this gradient is created by the movement of ions like sodium and potassium against their concentration gradients. So let's take, for example, potassium levels here inside of your cell is a much higher concentration inside than out. Now, if this was just allowed to diffuse, it would be moving from the inside to the outside just by diffusion. But our cells actively pump potassium inside. So it's working against the concentration gradient, so it takes energy. And then it's the opposite for sodium. Outside of the cell here, there's more sodium than potassium, and our cells are actively pumping it out against its concentration gradient. Now, if you'll notice, this is actually working in 2 ways. There's this chemical force, so that ion's own concentration gradient, and then there's an electrical component to this. Again, the inside of the cell slightly negative, outside slightly positive. So that's contributing to the differences in this membrane potential on either side. So that sodium-potassium pump is a key contributor to that electrochemical difference across the cell membrane. This proton pump also contributes to that. So here we see this proton pump pumping protons, hydrogen ions, which have a positive charge, from the inside to the outside, again contributing to the outside being more positive than the inside. Now, one of the interesting things here, this is going to become really important when we get into metabolism. You've created this gradient. You can actually use this as potential energy, because these hydrogen ions actually want to move back down their gradient. We've spent energy to move them out, but they sort of quote unquote want to move down their concentration gradient, or they're just going to naturally. And if that potential energy can be harvested, it can be used to do things. So let's take a simple example of that. So hydrogen here is going to diffuse down its gradient, and it's going to do so through, in this case, through this co-transporter, or symporter. And what's it going to take with it is sucrose. Sucrose, in this case, is being moved against its concentration gradient. So this is active transport over here, and it's using the energy from this hydrogen ion gradient. So we're pumping hydrogens to then move them back in with sucrose against its concentration gradient. So this is an example of co-transport. You need to first get the hydrogens out, and then the second part of this is moving them down their gradient, using that potential energy to move sucrose into the cell. We see that here in this example as well. So here's our sodium-potassium pump. We'll see it in a little more detail in a second. But here we see the high sodium outside of the cell, and we can use that concentration gradient. Sodium can help drive the movement of glucose against its concentration gradient from outside to the inside of this particular cell. And so here's our sodium-potassium pump again. We're seeing it a little more closely. We can start with 1 here. So sodium gets loaded up in this protein, this transmembrane protein, and it's not going to just move down. This is moving it against its concentration gradient. See high concentration outside the cell. So this isn't going to happen just naturally as a passive process. You need energy. So here's ATP coming in. This protein gets phosphorylated. It changes shape, conformation, and pukes out these sodium ions into the outer space around the cell. Now this protein can accept a couple of potassium ions. And again, we're still moving it against their concentration gradient too, and they move a couple into the cell. So you're getting kind of 2 for 1 here. For each ATP, you can move 3 sodium ions out and 2 potassium ions in. And so you can start to see how these processes are very much connected. You've got this electrochemical gradient across the cell membrane that's contributed to by different ions that are pumped actively against their concentration gradients. That potential energy of one thing on inside or outside of the membrane can be used to move something else against its concentration gradient. And then also we're looking at this sort of one bit of a cell at a time, but what might be happening on one surface of a cell? Say these are intestinal cells here. Actually, this is an example of kidney cells. So glucose and sodium being taken up into the cell. And the glucose is actually being moved out the other end, the basal part of the cell here. And the sodium-potassium pumps are actually on this part of the cell too. So where on the cell these are also matters. There's different conditions, say on this, where this part of the cell is and this part of the cell in the kidney, for example. All right, so let's start to put some of this together. So given the figure below, what experimental treatment would increase the rate of sucrose transport into the cell? Looking at the picture, you're gonna have to figure out where is inside the cell versus outside the cell. There is a way to figure that out. So given the choices here, what's the right answer? All right, so that one, maybe takes a little bit of thinking because there's a few concepts going on here. You have to recognize that this negative, showing the little negatives here, that's going to be the inside of the cell versus the outside, which is slightly positive charge. And here we see hydrogens being pumped. So if we can get more hydrogens on this exterior part of the cell, we can pump more sucrose in if we can increase that gradient of hydrogens. So one way to do that would be to decrease the pH, right? So that would on the outside here, which would mean if the pH is decreasing, we're actually increasing the hydrogen ion concentration. So that's, taking a few different concepts you've learned from so far and starting to put them all together. All right, so let's move some things in like big amounts here. This is called bulk transport. And so there's 2 general kinds of bulk transport, endocytosis and exocytosis. So endo means coming in, exo means going out. And we see both of those here. Here's an immune cell here taking up some bacteria. So this is endocytosis, taking them in. Here we see lysosome fusion, breaking down of those little bacteria, and then exocytosis of the waste being released out of the cell. And so endocytosis is split into a couple of categories. One is phagocytosis. So phago or phagi means eating. And so this is sort of larger things being taken up by the cell. And I'll point out here, look at this bacterium. It's being encapsulated by this cell membrane. and then it ultimately forms a vesicle around the thing. So it is taken up, and all this, whatever liquid was out with this bacterium outside of the cell, is now inside here. Pinocytosis, pino means drinking. And so this is really just taking up smaller things like solute in batches like we see here. So again, taking up these molecules, and now they're contained within this vesicle. A special kind of endocytosis is receptor-mediated endocytosis, and this is very specific. So it's not just, well, whatever happens to be floating around out there. These receptor proteins will bind very specific target molecules, so only those certain things are going to attach, and then those are taken up. So this won't just take up any old thing. It's got to take up these little green target molecules. How about exocytosis? It's just the opposite of what we've seen so far. So here's a waste or secretory vesicle. Maybe these are proteins that need to be released outside the cell, not just because they're waste, but something else that's important, a signaling molecule, all kinds of things. But notice the cell membranes will fuse here, and what was the inside of this vesicle now becomes continuous with the outside of the cell, and poof, out go those secretory products. All right, so last question here in this session. What best describes bulk transport, given the options here? I always like to ask this question because it makes you think about what's truly happening here. And if we look at our choices, you might have got them in a different order here, but all these other choices are all about molecules of whatever kind actually crossing a phospholipid bilayer. But if you look at what's happening here, this bacterium, for example, it hasn't crossed a lipid bilayer at all. It's just been internalized in this vesicle. And so that's a key thing when we think about bulk transport and how these membranes are dynamic, and they can fuse and form vesicles and release things in exocytosis. So make sure you have a good handle on that. And we'll see you in the next part.